Okay, so John, before we jump in, can we just have a chance to introduce our two new panelists? Manuel, do you want to introduce yourself briefly?
Yes. Okay. Emanuela Taioli, I'm an epidemiologist, the Associate Director of the Cancer Center, Mount Sinai, New York.
So my name is Yatabe. Hello, can you hear me? Okay, so my name is Yatabe. I'm a surgical and molecular pathologist working in National Cancer Center Hospital, Tokyo, Japan.
Okay, and John, you've already introduced yourself. Before we jump into the series of questions that we developed, I think it's worth just running through and seeing if any of our panelists have any burning comments they want to make about what we just heard. Does that sound reasonable? Is that okay, John?
That's fine.
Yeah. So Hillary, you can say whatever you want about what you just said, but we might start. Yeah, we might start with Emmanuel.
Very stressful. The microphone is very stressful. Yeah, so one comment that I thought while Hillary was presenting and in general is that you really need to position your biomarker where you want to obtain an improvement in your process. And I was thinking, while Hillary was talking, for example, even after the first cancer screening detected that is a stage 1, there is no therapy. Well, biomarkers would be even good to do an early detection of a second cancer. So there are several places, and depending on where you want to place your biomarker, the requirements are different and the expectations are different. So we need to define that together with study design, I think.
Yes.
I would like to make a comment on the biomarker research because the biomarker to detect early lung cancer is quite different from the biomarker, for example, ctDNA, which was now performed in clinical practice, because most of the ctDNA or blood marker is associated with a stage. So that's because the tumor cell, that biomarker is released from the tumor cells. Therefore, the biomarker is associated with the tumor burden, the volume of the tumors. Therefore, the early detection biomarker is quite different from the others. So that's my comment.
Okay, thank you very much. John, do you want to start out with the questions?
Thank you very much. I was going to just add to the sort of overall comments. I mean, Hillary's given us an excellent overview of where we stand at the moment, and hopefully the protein biomarkers will, in fact, prove to be very useful. But in my mind, we need to focus on where biomarkers will actually have the maximum clinical impact. In other words, the biggest bang for bucks, the bang for bucks. Is it going to be early detection? Is it actually going to be identifying nodule malignancy? Or actually, is it going to be identifying individuals that are going to go down various therapeutic pathways? And maybe that will actually help us also to look at the type of funding that in the future that is required. I mean, the Integral project has actually attracted an enormous amount of money, which has enabled the project to move forward. But Volker has actually demonstrated that is only half of the question. The other half is that we need to focus on how are we going to get industry involved and how do we actually take it through the regulatory pathways. So that was the sort of overview I got from the various talks. But going to the particular questions, if we actually stood back and said, what is ready for clinical application today? and what planning is required in the next 3 years? That's the first question I would like to put forward.
Fred?
To be honest with you, John, I think an important comment was made here previously by Emanuela and others. We need to clearly identify what are we talking about in terms of biomarkers. What is the purpose of the biomarker? And I think we are going back and forth with diagnostic biomarkers, risk biomarkers, risk after surgery. We need to clearly separate what are we looking for. Point two, there is a lot of studies going on and has gone on for years. What we are missing is what Volker is talking about, a strict process where we have a strict validation, clinical validation of the biomarker panels we are talking about. And third, we might still lack the real biomarker because we don't know, and now I'm talking in the screening setting, we don't know the biology of the screen-detected nodules. We know biology in terms of radiographic appearance and the cores. We talked about it yesterday. David pointed out the importance of seeing the time frame where the nodules grow. But again, I would emphasize what I'm missing is the biology of those nodules. Remember again, what we learned in advanced disease and what led us to personalized therapy was we learned about the biology of the tumor.
So, Fred, I would like to pick up where you're talking about and put a sharper focus on it. Because, you know, I'm very tempered by listening to Sudhir Srivastava talk about every 5 years about how they spend another $60 million and have nothing to show for it. And that it requires a analysis of strategy. Now, you could do a biomarker analysis for early detection and try to compare it and stuff like that. And you have to study, it's incredibly difficult to study because you have to study 100 cases to get one cancer, you know, depending on the context and stuff like that. And it's just very difficult because the bar is so high because CT is so good. The place where CT doesn't work emerges from the analysis of Claudia's 25-year data. And she has success with 80%, 81%. And that means there's, you know, roughly 1 in 5 tumors that despite, you know, the performance of CT and stuff like that, wind up being the wolves in sheep clothing, you know, the editorials you see periodically. So if you applied your strategy of trying to work out the biology of those resected primary cancers, screen-detected primary cancers, and looked at for the ones that correlated with, you know, aggressive behavior, either, you know, and I don't know whether it's going to be persistent, residual tumor in the bed, or it's another tumor occurring in that same individual 18 months later metachronously. But if you look at the 20% of failures, instead of having to study the hundreds of millions of people at risk, you're down to the 1 in 100 that actually has a cancer case. And within that cancer case, you're going to look at the failure of CT screening because identifying targets and drugging those targets or whatever is something that we're very good at. So working out the biology in those wolves in sheep clothing is an incredibly high reward opportunity amongst all the other options that we've been talking about. And it would fit with your kind of tissue is the issue, figure out what's going wrong, and drug it. Any ideas? Hillary, what do you think about that as an epidemiologist?
Well, I mean, it's interesting because it's exactly the opposite of the approach that we took. And it doesn't mean that one is right or wrong. I think they're complementary. One avenue is to, tissue is the issue, is to take the tissue, learn about the biology, use that to inform what biomarkers you think are going to work, and then go sort out whether they work. We've done the opposite, which is sort of completely agnostic, fully data-driven, scan everything and figure out what's predictive with sort of zero biological information feeding into it. But both can lead to success, in fact, but I kind of think both should be considered. And as an epidemiologist who, you know, sort of works with population data, I have to admit I'm a bit biased towards the second one, because you find things that you wouldn't have anticipated. I mean, the proteins that we identified, some of them seem to be related specifically to the tumor, but others, we're not quite sure what they're doing, but they validate.
Yeah, thank you. Emmanuel, your comments?
Right? Okay. Oh, yeah. Okay. So I think I completely agree with the idea that the tissue would be, the biology of the tumor is really important, because if we say what biomarker is good, against CT scan, nothing is good enough. I mean, then you lost. So, but the other thing is, for example, if there is over the follow-up no adherence to the screening program for whatever reasons, then maybe a biomarker can help filling in the gaps, because that's better than nothing. And maybe the biomarker is not as good as the CT scan, but it could help. So there are situations that you need to see in real life.
Yes. Dr. Yatabe?
And also that we pathologists know the
Can we speak a little louder? I think.
Is that okay? So because we pathologists know the biology of the lung cancers. For example, one article reported the ground grass nodule, or that was followed for 1 or 2 years. So how and that tumor's tissue revealed some sort of driver's mutation. The KRAS mutated tumor, GGO, is sustained as a GGO for at least 2 years. However, EGFR mutated tumor progressed into the core portion. So that's kind of the biology can be applied to the early stage the detection. So we know the biology. Biology could be quite important. I agree with Fred, and that's kind of the things should be applied to the early detection as well.
Thank you. Very, very thoughtful. Volker, you had a comment?
Yes, absolutely. Thanks for the opportunity. Let me just one step going back to the immediate comments and just point out that it really hurts me when I hear that, you know, everything is made, but then not for commercial purposes, because I would, you know, just to be a little bit provocative, say, how are you going to do regulatory? How are you going to translate? How are you making sure this actually becomes a game changer in the real world? Now, if we do this sequentially, and this is the current approach, it takes very, very long. So it's really something to think about from the way we collaborate. And then listening to what you said, I fully agree. There is CT issues, there's CT bias, there's population bias. I don't think one is right or wrong. The question simply is, how do we get successful? And all of those have a right to play, but we're not succeeding on any of them. So I would rather focus a little bit on why are we not succeeding than fighting over, do we need to solve nodules? Do we need to identify risk? That's a second question from my perspective.
Yeah, but I'm sorry, was there somebody who had a comment? Okay. Volker, I think the idea that you could take several sites, big volume sites, and do discovery on some systematic way of the next 100 cases coming from 10 different institutions. And as Dr. Yatabe just talked about, look for druggable components in cases that were that wound up being early progressors or whatever, the people that were not doing well after a curative stage 1 resection. I think that's a very finite thing, and you already have a number of targets that could be almost immediately applied if you had KRAS or some of the other actionable mutations or perturbations that Dr. Hirsch works with. So I think there's gold in those hills, and I'm surprised we haven't done that yet.
Well, we are on the way with this. I think Charlie Swanton's group in UK is leading the effort to characterize bronchial epithelium and the preneoplastic changes. What does this PM 2.5 leads to? We know the pollution factors, but we don't know what does it lead to in terms of carcinogenesis. But we are starting to learn that some of those driver mutations, they are present already in a very early stage of development. But there might be factors that trigger their potential to be a driver mutation. But that is a direction we are going into these days to learn about biology. So we are hopefully coming there, and hopefully it will lead to earlier prevention.
So I appreciate what you're saying, but what I'm asking is, why wouldn't you let the biological information from a failed screening resected cancer help you identify things that are very...
I agree with that.
Yeah.
I agree with that totally.
Okay. Dr. Kelly, you look like you have something to say.
So I think this is a fascinating conversation that we're having, because in our world, it really is all about biomarkers and how do we continue to slice the pie to get, I think, Jim, at what you're talking about, that optimal care for those small groups, as you're just talking about, that small group of those stage 1 patients who we all have that surprisingly met out at the next scan. You've resected them, and literally 3 months later, we all have these types of patients, although rare, that met out. And those are the patients that we really need that biomarker for in early stage disease. But this is something that we do all the time. And as you know, we keep slicing the pie even thinner. And then when we slice the pie, how does it apply to early stage disease? I was thinking, you know, we're never going to do studies to show that a ROS1 inhibitor is actually beneficial in early stage disease. It's just too small of a number. Now, maybe if we had a really great international collaboration, we could do that. But this is the big question in early stage disease. Even now, these are so rare that for a company to want to spend millions and millions of dollars to prove that a ROS1 inhibitor works in earlier stage, just like EGFR does and now ALK, it's very, very hard. And we keep asking the same question without any answer to the question that we're doing. So I'm just fascinated by the conversation. I certainly agree that ctDNA in early stage is a completely different ballgame than ctDNA in late stage. And as we are trying to grapple with the 10th edition staging, which we'll talk about tomorrow, how can we maybe collect blood, be a little bit more innovative in staging? Because we have such an international opportunity, can we actually collect blood in the staging in the 10th edition? We're debating that so that we can collect it. And when that FDA approved test comes, can we look at it in all those samples in a uniform way, that would really be, to me, the step forward.
Yeah. So what do you think about if someone came up with a panel that they routinely run against resected stage 1 screening cases? You pick out your high risk targets and stuff like that. You create a panel that 20 institutions or whoever wants to runs it, and they get reimbursed if they send in the data.
So if I'm correct, Dave Jablon's had that panel. Fred, you know what I'm talking about.
Yes, yes.
And Jim, you know what I'm talking about, developed a long time ago, and that has never really, it's approved as a laboratory test, but it's never taken off. And we tried to do twice a prospective trial to look at if you had the high risk features from that panel, you got chemotherapy. If you the intermediate to high, you got the chemo, and then if you were in the low arm, you didn't get any treatment. And that never, ever was able to the accrual was so poor on that trial. And I think we tried it 2 different times. So I don't know how to get us there to having a panel that works. And as long as you have to have an action item, not just an observation.
Yeah, but I think about like the family genetics programs, in which they get rare patients and they work up the families and stuff like that. They figured out some very efficient ways to do this. I think coming up with an efficient way to get that resected tumor tissue from these resections so you could test the David Jablons more completely, and the Jablons 2 and 3 and 4, whatever the candidates are out there, there's no reason why that couldn't be done now relatively efficiently. Saying that, for any institution that has these cases, they want to do this panel, whatever you describe it, experts do that. That could be done. David,
It's what to do with the results.
Pardon?
It's what's to do with the results of getting the panel done.
You look for consensus targets that turn out to
It's
be
the gray
hotspot.
area. It's the intermediate. So in David's, you have the high, the intermediate, and the low. The low and the high, those make sense. That intermediate area, patients did not want to go on to cytotoxic chemotherapy for an intermediate score. They'd go on it for a high score, and certainly you wouldn't put them on for a low score, but we have that intermediate that makes this gray, and therefore that was one of the reasons.
No, I think there's real world considerations, but if you could do the analysis and it turned out that some targets popped up that you had relatively benign interventions for, that could work.
Yep.
David,
Jim,
you
can
don't
I
care
make
about
a-
this topic at all.
Jim, can I make a comment? Jim?
Put it to a vote. Yeah, go ahead.
Okay, Jim.
Oh, no, just a second. Okay.
Jim,
John, your comment, then we'll go here, and then we'll go to Professor Yankelevitz.
can I
David,
just come
John.
back to what the second part of our question was, planning required for the next 3 years? If we actually look at the investment of the ERDN over the last, let's say, 5-10 years, it's been enormous, but actually its output has been, let's say, poor. And I agree with Hilary. I actually think our whole approach has to be data-driven, but actually, do we need to reconsider how we actually develop our biomarkers? And actually, I think they have to be not only the data-driven, but actually within a clinical trial approach. Because if we actually think of our main sort of comment with Agile, we want to be thinking of the big 3. We're thinking of smoking-related diseases. So we shouldn't just be focusing on lung cancer. We should be actually thinking of cardiac disease, COPD. We have this risk population, and we have the opportunity to ask multiple questions, and thereby hopefully bring in multiple sources of funding and maybe industrial funding. So when we actually say, what should we do in the next 3 years, I actually feel this is something that Agile can really lead, because most groups are very focused on one specific area of CT screening. But if we actually broaden it out to the big 3, and we actually say we have an overlapping interest, lung cancer, cardiac disease, and COPD, I actually think we could actually change the whole paradigm and the, let's say, approach that we take over the next 3 to 5 years.
I think it's an important point. Thank you, John. Yes? microphone? Yes.
If you want to have biological explanations, in terms of the biological markers, just share the previous screening experience in breast cancer screening as well, because you can always, look at the biomarkers between the screening detected and interval cancers, for examples, and even non-participant. In the old day, we couldn't do that. It's so difficult. And now you got so many the protein and omics data, so they can complete the friend talking about the biological pictures. So I think this is very important, and you can always use the nested case control studies to samples, only few the interval cancer or non-participant, and also screen detected compared with even the normal for the polygen. So it's not difficult today, even the AI. So this is first comment. I think here, know that you probably plan to do that. You can make some comment. But the second comment, I think it's very difficult because these biological markers always very dynamical. So you never know when the measurement is correct to reflect the tumor carcinogenesis or the progressions. So dynamical is so difficult because screening has schedule. I know you have a time dependent, but the biological also time dependent. The two time frames are not matched. So it's makes the AI analysis very, very complicated. So I want Harry, if you can make some comment about that. And that could be one of the solutions. I heard this morning, Jill, you're talking about the volume change. You're talking about so many, difficulty for measurement change. But how about to use the biomarkers in parallel with the change with the nodule? Then they can feel these biological possibilities.
Yeah. So you add another layer of analysis. It could be done very well. David?
Well, I wanted to sort of circle back a little bit to one of the comments that Professor Yatabe said, who, by the way, reminded me that he was the pathologist at the I-ELCAP conference in Nara some 20 years ago. Wonderful. So that he is familiar with our organization. He talked again about, and this is the topic that I was sort of alluding to, those markers that are produced by the tumor itself. And this is, to me, this is a distinction that I want to just get your opinion about. When you look at bronchial dysplasia or something, that to me is a marker of risk. It's not telling you the tumor's not producing it, but it gives you an idea that a tumor, this person is likely to get a tumor. Whereas when something's produced by the tumor, and it shows up in the bloodstream, and that tumor's already been there for 20 years. It's been growing. We just may not see it. So it's a little bit, to me, this is a very different concept than a marker of risk. Even though you're not seeing the tumor, you're seeing something that's telling you there's a tumor that's hidden, but it's just slowly growing, or it's in the background. And to me, these are sort of different concepts. And so I'm just wondering how we should think about this, because to me, one is more of a diagnostic test, and one is more of a true risk test. Smoking is a risk factor. Dysplasia is sort of a risk factor, some genetic alteration. But when you're talking about something produced by the tumor that we don't see the tumor yet, to me, this is much more, diagnostic issues, so, and that's kind of what you had said. So can you maybe further elaborate on that?
Yeah, because so I mentioned about the error detection is quite different from the.
Can you speak a little louder, please?
Okay, how about that?
Yeah.
Is that good? So because the biomarker early detection for early detection is quite different. However, if the biomarker which were sustained or very, very stable and the large amount of release from the tumor, even in that small size, it's independent of the size. So that could be a biomarker for early cancer detection. So however, most of the current biomarker is associated with in the blood is associated with tumor burden. But probably Hillary's the study design can be detected with such a stable early detection proteins in the blood. So I think
Yes. So we have Volker, then we have Fred.
So David, I respectfully disagree with you. The biomarkers in the tumor, many of them, as we today know, are already persistent in the pre-neoplastic scenario as well as even before that. And that is demonstrated for EGFR, for KRAS in Charlie Swanton's studies today. Going back to the EGFR story, today EGFR is, you know, the landmark of personalized therapy for lung cancer. But people forget that before the mutation were detected, we detected several years before that, EGFR was already, and Karen, remember that because it happened in Colorado, we demonstrated EGFR was there already in an early pre-neoplastic situation. And we demonstrated that as more dysplastic the epithelium became, as more EGFR did they express based on protein, based on gene copy number. And Charlie Swanton today demonstrated that pollution can affect EGFR mutation even earlier. So what you're talking about, the tumor biomarkers might have existed early on.
Yeah, kind of field carcinogenesis type things. Volker, you had a comment?
Yeah, just briefly on this one, I fully agree with Fred Hirsch. We see it, at least in our biomarkers, that they're not completely correlating when you take the tumor out. So there are system responses as well. And given the complexity of biology, we can be sure it's all of us. So there is no right or wrong, as discussed earlier. But let me come back to what John was saying, because we are here today because there were early studies on CT screening some 25 years ago. They generated the data, they triggered the clinical trials, and even the clinical trials are 15 years old now, and we are still using them. And now, with the new implementation studies, there's the second phase of big trials. And it's a unique opportunity now to do better, to actually develop something where we come together in a learning system, as you described, find something, go back. But the only way to go back in time is to use either biospecimens or existing data. So that's what we need to optimize. And with what I sense here, the gap between the different approaches between research being interested in certain questions and industry looking at different questions, it's really difficult to get these situation of the implementation trials used optimally, right? We looked at urine biomarkers. There's no biobanking in the world of a meaningful quality that we could get access to. We looked at population databases. They are fantastic ones, but actually for regulatory purposes, they're not screened populations, so they're not acceptable. Blood is leading the field just because of availability. That's why the others lag behind. And now we have the chance to do it differently. And I heard it earlier in the sessions, biomarkers are just not available in clinical routine. CT scanning was available before the whole story started, but the biomarkers are lagging behind because it's a novel technology that is clinically not available. So it's really the time is now to act on these studies to do it differently. And we don't have the model yet how to do this.
Okay, thank you very much. I just want to check, do we have any questions from the Zoom populations?
I have one question.
Yeah. Okay. So please let me know, but go ahead.
So thank you for this wonderful discussion. My question is, we have the chance to have someone from the World Health Organization here. And my question is, what is the overreaching goal of the World Health Organization? Is it to screen everyone for every cancer starting age 30 or 40? And to be more specific, now we can maybe start to screen everyone, even never smoker. Do we have biomarker in terms of proteins for starting to test in a never smoker population?
No, I would say definitely not. I would clarify first that I don't speak on behalf of WHO and I don't speak on behalf of I-ARC. However, WHO currently recommends screening for breast, colorectal, and cervical cancer. There's no formal recommendation from WHO for lung cancer screening. There's a very important development happening here because at I-ARC, where I work in Lyon, we are just beginning the process to publish a handbook on lung cancer screening. And the I-ARC handbooks of cancer prevention are the formal evaluation of cancer prevention interventions. So the last one was on oral cancer prevention. They exist for colorectal cancer screening, cervical cancer screening, and so forth. So over the next 2 years will be the handbook on lung cancer screening. And it's very, very important from a policy perspective. So that will be sort of the evaluation that will provide an answer to your question at the end.
Thank you. Rowena, did we have any questions on? Can we get a microphone for you, please? I'm sorry. Thank you.
The first one is for Hillary from the first presentation, comments about urine tests. And the second question was, are these
Can we get a little louder, please?
Are these biomarkers going to put more pressure in the health system if more people is opt into screening programs?
If more people
Opt into screening programs.
opt in, there's many different ways we can use biomarkers. One of them is to actually reduce the number of people who are screened. One of them is to keep the number the same and find more cancers. Another could be to screen more people. It completely depends on the health system context. So I would say not necessarily.
And any comments about urine tests?
Can you say it again?
Urine tests.
A urine test. The blood data are much more interesting than the urine data. There are good urine samples from some of these population cohorts, but as Volker alluded to, they might not be accessible to industry.
Okay.
So John, for an information, we talked, John Field mentioned we should look for a biomarker for the big three. And I don't disagree with that at all, but I think a huge focus these days is in never smokers. And this morning, I received an email from Marina Garasino, and she wanted to make, based on our international collaboration during COVID in TerraVault, she wanted to make a family history database on an international basis. Might be something I-ELCAP should be involved in also. But getting together and have a family history database with blood and genetic markers would probably be a good pathway forward to learn how to deal with never smokers. And that is, of course, based on the Taiwan group's the talent study. The family history is important. So hopefully we can collaborate on that part of it.
Dr. Wang.
Just a brief comment. Actually, the talent sample, part of the talent sample already sent to Charlie Staunton's and also Regine to verify whether this biomarker is also workable in non-smoking lung cancer hopefully we can get the data soon.
Okay, other burning issues? So this is one of the questions, and we've covered part of it, but not all of it. Is there better statistical mathematical tests, especially in the context of describing risk markers for screening? And that's a question. And then the question that I have from some of the presentations is, how do we use the intersection between some of the emerging information we're getting from the biomarkers with the modeling study that SysNet and others are doing to kind of allow us to develop more informative results? Hillary, do you want to take that?
The first question was, are there better statistical methods compared with what?
So is there any methodology challenge that, you kind of say, we should do it this way, someone else says we should do it that way?
I would say no. Novel statistical methods are usually not what we need. Better study design and better data and samples going into the whole machine is usually what we need. And the second question, I'm so sorry.
How do we interact between the biological information emerging with the SysNet modeling?
SysNet, yeah. This is difficult to answer succinctly because our discussions with SysNet have been really quite interesting because we're trying to parse out exactly what information they need about the biomarker performance in order to adapt their modeling and marry the two together. And it's really not simple. But for example, a question we have is, who the heck do we test? Let's say we have a great biomarker for pre-screening risk to inform the decision of who to screen. You do not need to test everybody because some people are so high risk that they will never change their decision. Other people are so low risk that they will never change their decision. We need to test the people in the middle most likely, but who exactly? So in order for SysNet to sort of work this out, we have to iterate with them and sort of provide the pieces of information that they need. But the good news is that because of the way that we designed our study, we can sort of come up with anything that they might ask for.
Yeah. So on that point, Harry was showing earlier how they did this a little bit with SysNet. And it was interesting to me. One of the things they tricked upon was the fact that at 15 years from smoking cessation, they thought that they could potentially stop screening those individuals, et cetera, and their modeling data supported it. In point of fact, the American College, the thoracic societies and stuff like that, when they looked at it, I forget what the fraction is, but basically, 40% of lung cancer cases occurred in individuals that have been, remotely, had stopped smoking in the remote past. So that SysNet model may work in some validity, but it also kind of ignored the reality of the fact that they were missing a lot of the potential target lung cancer cases.
Indeed, and I don't think it was the model itself, but the scenarios that were fed into the model. So the scenarios that it was asked to evaluate were those with a 15 quit year threshold. But it doesn't mean that you couldn't evaluate other scenarios with a well-constructed microsimulation model.
Okay, so Emmanuel, did you want to say something about that? Thank you.
Sorry. Yeah. Now, I think it's because of their model, partly because they're dealing with the fact that there is not one way to define risk. There is a very individual component that we haven't caught yet completely. And there is where biomarkers could be helpful in a way, because their model is purely mathematical. They just put their data in and they model different strategies, but they don't look at the individual risk as a variation. They don't characterize the individual risk more.
Okay, I'm sorry. Ilya?
You had a wonderful slide that showed where the biomarkers can actually play the role, which I think eventually will result in the same type of eligibility criteria. And that's just a comment. But I also have a question. With those tests potentially becoming more available, more sensitive, cheaper, do we also, or do you also take into account the potential burden on the patient? Something comes out positive, it does not necessarily mean that there is cancer, there might be cancer. How does this all play together in your study design?
Absolutely. We are always thinking that there is patient burden. This has to add something, right? And I sort of discussed that. And perhaps what you're getting at, I mean, it's an interesting point, and it came up recently in designing this acceptability study that I mentioned, which is slated to launch later this year in Kentucky with Mike Gieske, who some of you know. And so we're sort of working through the communication materials. And what we're doing in this study is we're taking people who aren't task force eligible, but they're almost task force eligible. And we're testing them with the biomarker panel and evaluating their absolute risk of lung cancer. And for those who sort of cross a threshold that's equivalent to the task force, we refer them to LDCT screening. And we realized that when we deliver that message, we have to distinguish between your risk is higher for lung cancer versus they think this is a test that says they have lung cancer, you know, and for a participant, it's quite complicated. So what we're planning to do is sort of ping pong with patient groups, and not lung cancer patients, but cancer-free people, because that's who we'll be dealing with, in order to make sure that we don't cause sort of unnecessary stress because of the way that people might misinterpret what a blood test means.
Thank you. One last question.
No, that is not a question. That is a comment. I glanced the program here, and I think many in the audience believe that 5-10 years from now, the screening scenario will include a combination of radiology, biomarkers, and might be some other things. But and here in the program here, and that is appreciated, of course, that you have put biomarker in the program with 1.5 hours. But might be the Agile consortium should think of including a biomarker consortium with to focus prospectively how biomarker approaches can be integrated in future studies for screening for lung cancer. I cannot talk for cardiac disease, that is not my specialty, but I'm sure there is room for that too.
Yeah, and Fred, I think your comment is about molecular biological markers. You know, there's people in this audience that are working right now intensively on novel imaging biomarkers, angiogenesis, all kinds of other signatures that are emerging, you know.
Radioisotopes.
No, I'm just talking within the CT world, because we haven't talked much about the photon counting, but the information load that we're going to get from that generation of scanners is exponentially higher than what we've had up until now. And the hidden information that you could extract from that with our modern computational tools is staggering. Sean, do you want to help me on this in terms of some of the information that might coming out from some of these new biomarkers? Do we have a microphone?
Nodule shape and texture. I mean, those things can be looked at now, but I think with photon counting, the resolution will be a significant step forward. The other points I think that were discussed earlier too, involving emphysema and cluster analysis of emphysema, those are all developing and emerging tools that will probably have impact as well.
And there's a whole line of that. to the nth degree coming out of the cardiovascular world, imaging world as well. So we have a wealth of new things that we can weigh into. David, did you want to have the last word?
I'm going to have the last word, but it has nothing to do with biomarkers.
Oh.
The last word has to do with Matt Sham is here. He's our official
Photographer in chief.
For Agile, and he is eager to take a group photo of this very handsome audience. And
Okay.
panelists.
Bruce, did you, last word, very briefly.
We modeled this and it's a function of the improvement in sensitivity. I don't know.
Okay, on that we'll close. Thank you very much. Thank the panelists and presenters. Thank you, John. Thank you, Volker.
Thank you.